This chapter, we are going to continue to look at still on chapter 7, which is on immune system. Last lecture, we have looked at roughly the overview of the immune response. Okay, immune response. So as you know, immune response, it has three line of defenses, which is you have the first line defense, second line defense, and third line defense. Okay, so for first line defense, we call it as the external barrier. Okay, so for example, you have your skin, okay, which is it is maintained in acidic condition. Okay, and then you have, for example, um, mucous membrane. Okay, mucous membrane, and also the secretion from the mucous membrane. Okay, so example of secretion from the mucous membrane, you have the tears. Okay, so tears, inside the tears you have lysozyme. Okay, that function to destroy the cell wall of the bacteria. Same goes to your saliva. So your saliva, the food that you eat is not 100% clean. Okay, it may contain pentogen. So the function of the saliva is as for external defense. Okay, to digest if there is any possible contamination of bacteria. But if the bacteria or the pathogen manages to enter into your stomach, then inside your stomach there will be secretion of hydrochloric acid. Okay, so the function of hydrochloric acid is to kill the bacteria. Okay, apart from another function of HCl is to activate the enzyme pepsinogen, so that it is converted into pepsin to digest protein. Dah belajar kan digestive system dulu masa kat sekolah kan. So kalau perut ni adalah untuk pencernaan protein kan. Sebab apa, dalam perut ni ada enzim pepsin. Okay, so in order to activate the pepsin, it needs the hydrochloric acid. Okay, so hydrochloric acid basically it is also to kill the bacteria. Okay, that enters with the food that you eat, and then that is the first line defense. Okay, and then the second line defense is where it involves, for example, white blood cells that have the capability of engulfing the bacteria. We call it as the phagocyte. Okay, so you have several type of phagocyte. You have the neutrophil, you have the eosinophil, you have the macrophage, you have the dendritic cells. Okay, so these are the phagocyte that will engulf the bacteria, okay, or the pathogen. And then you have also the second one is antimicrobial protein that we are going to look at today. And then you have the NK, the function of natural killer cell. And then you have inflammatory responses. Okay, so those are um, the four component of second line defense. So for first and second line defense, the mechanism is much more rapid compared to the third line defense. Okay, so maksudnya rapid, maksudnya it is there. Okay, it is there to protect your body against any possible entrance of pathogen into your body. Okay, but if the pathogen manages to penetrate first and second line defense, it will encounter the third line defense, or we call it as the acquired immunity or adaptive immunity. Okay, so for this third line defense, it will protect your body against invasion of specific pathogen. Okay, specific pathogen that are harmful to your body. Okay, that may cause disease. So why do we call it as specific? Because it involves white blood cells. Okay, specifically it involves a lymphocyte that on its cell surface it has receptor. Okay, it has receptor. Okay, so this receptor will bind to the antigen that are present, especially at the surface of the virus, for example. Okay, so kalau orang biasa kan tengok macam contohnya gambar virus COVID-19 kan, so ada pacak-pacak kat luar tu kan, itu that one we call it as antigen. Okay, so the function of the lymphocyte, okay, it has many receptor on its cell surface. So if the lymphocyte that has receptor that matches to the antigen, to the pentogen antigen, okay, so it activates that lymphocyte. Okay, so once the lymphocyte is activated, it will multiply into many copies, into many cells that has the receptor for that pentogen or virus. Okay, so that will activate your immune system, okay, your immune cell against that specific virus. Okay, so once this lymphocyte is activated, it will form two colonies, okay, which is activated colony during the time of infection, and another colony is the memory cell. Okay, so this memory cell will be stored in your lymph nodes. Okay, and then if your body is infected by the same virus in the future, so you will not get sick anymore. Kan, kalau first time kena infected by a new virus, you may get sick. Okay, so itu cara badan awak nak build up sistem pertahanan lah. Okay, tapi once your body is activated, or your immune response is activated against that virus, when you are infected again in the future by the same virus, you will not get sick. Okay, so that is the function of your immune system, the third line defense or the acquired immunity. Okay. Okay, so now going back, okay, into the second line defense. Kita dah cover hari tu sampai ni secretion. Okay, so now we are going to look at internal defenses. Okay. Ini kita dah masuk ke belum? Phagocytic cell ni. Boleh dengar tak saya cakap?
Belum, madam.
Belum cover Yeah. Okay, so internal defense. Okay, so again, internal defense is second line defense. Okay, second line defense. So ingat innate immunity, first line and second line. Okay, first line is external barrier. Second line is internal barrier or internal defense. Okay, this is innate immune response. Okay, innate immunity. Okay, so internal defense, also known as second line defense. So it is triggered when the pathogen enters the body. So it has four components. You have the phagocytic cell, you have the neutrophil, you have the eosinophil, you have the macrophage, and then you have the dendritic cell. Okay, and then you have the antimicrobial proteins, such as, for example, interferon. Okay, and another one we are going to look at later. So this natural killer cell will kill infected body cell. Okay, natural killer cell. And then inflammatory response, it involves a series of reactions to occur when, for example, you have a cut. Okay, a cut that the cut, for example, may be prone to entrance of pathogen. Okay. So number one, phagocytic cells. So this type of defense is known as cellular counterattack. So this term you have to remember. Yes, cellular counterattack is it falls under second line defense, which is it involves the phagocytic cells. Okay. So because the phagocytic cell, we call it as the phagocyte, which is white blood cells that have the capability to engulf, okay, the pathogen through phagocytosis. Okay. So the mechanism of phagocytosis is the same as endocytosis. Kita belajar dulu kan endocytosis, exocytosis. Endo is to bring in substance from outside of the cell into the cell. In this case, the cell is the white blood cell. Okay, the neutrophil, for example. Okay, so phagocytes have receptor that recognize certain structures such as polysaccharides on the bacterial cell wall. Okay. So phagocyte engulf the bacteria and trap them in a vacuole. Okay, so ingat mechanism endocytosis. Endocytosis, one example of endocytosis is phagocytosis. Okay, so engulf the bacteria and surround the bacteria in a vacuole. Okay, so the vacuole will fuse with lysosome. Okay, so lysosome, you have to remember, it is an organelle. So inside the organelle you have enzyme, we call it as lysozyme. And also the lysosome produces nitric oxide, okay, nitric oxide, which is a free radical, and other gases that will poison the pathogen. Okay, so it poisons the pathogen first, and then the enzyme within the lysosome, okay, which is lysozyme, and other enzymes will degrade the bacterial cell wall. Okay.
Madam,
Yeah.
slide madam tak bergerak.
Sekejap. Sekejap. Okay. Ini dia. Alright. So that is phagocytosis. So I'll go through again. So phagocytosis is a type of endocytosis where the white blood cells, we call it as the phagocyte, that has the ability to engulf, okay, the pathogen. Okay, so it engulfs the pathogen because this phagocyte will have receptor that recognizes, or that is the pathogen. Okay, that is a pathogen. Recognize certain structures such as polysaccharides on the bacterial cell wall. Okay, so after the phagocyte recognizes, or that is a bacteria, so it will engulf the bacteria. Okay, and then after it engulf, it will surround the bacteria into a vacuole, and then the vacuole will fuse with lysosome. Okay, so inside the lysosome, it produces nitric oxide, which is a free radical, and other gases to poison the pathogen. And inside the lysosome, it also has this enzyme, which is lysozyme. Okay, lysozyme and other enzyme that will degrade the bacterial cell wall. Okay, so that is phagocytosis by the phagocyte. So this is the first of the second line defense. Okay, so if you look at this diagram, so here you have the white blood cell. Okay, so white blood cell or the phagocytic cell, okay, yang ada neutrophil, eosinophil, macrophage, and the dendritic cell has the ability to engulf the bacteria or the pathogen. So it surrounds, okay, the pathogen through the formation of pseudopodia. Okay, pseudopodia is cell extension here. Okay, it surrounds the pathogen through the formation of pseudopodia, and then it engulf the pathogen, okay, through the process of endocytosis. Okay, the whole process is phagocytosis. And then surround into a vacuole and then fuse with lysosome. So the lysosome contains tadi nitric oxide, poison that will poison the bacteria or the virus or the pathogen. And then after that, the lysozyme, okay, inside the lysosome will degrade the bacteria. Okay, and then after that, it has to excrete out, okay, the waste or the debris, okay, from the cell, from the white blood cell. So debris from pathogen are released. Okay, so this is phagocytosis. So ingat kat sini phagocytic cell you have 4 types. You have the neutrophil. So when you are sick, okay, when you are sick or demam ke apa ke, so neutrophil, okay, the number of neutrophil will increase. Okay, so sebab apa, dia adalah white blood cells yang akan datang dulu bila badan awak kena infected. Okay, infected by pathogen. Okay, so the number of neutrophil will increase once you are infected by virus. Itu yang akan increase dulu, bilangan neutrophil. Okay, so engulf and destroy pathogen. And then you have eosinophil. So it says here limited roles in phagocytosis. Eosinophil, okay, eosinophil has the ability to engulf pentogen through phagocytosis, but another function of eosinophil is to kill, multi multicellular parasite. Okay, parasite. Okay, multicellular parasite. Okay, so that's why dia cakap kat sini limited role, sebab fungsi lain dia adalah untuk kill multicellular parasite. Okay, it will secrete some kind of chemical that will kill the parasite, the multicellular parasite. Dia tak boleh nak engulf sebab eosinophil is one cell, kan? Dia tak boleh nak telan multicellular organism, multicellular parasite. Okay, so it will secrete chemicals that will kill the parasite. Okay, and then you have the dendritic cell. Okay, limited role in phagocytosis also, but it can engulf the pentogen. And the other one is macrophage. For these two cells, okay, macrophage and also dendritic cell, once it engulf a certain pentogen, okay, it will activate. It will involve in the third line defense. Okay, it will involve or activate the third line defense or the acquired immunities, because when these two cells, okay, macrophage and also dendritic cells engulf the pentogen, it will become a cell, we call it as APC, antigen presenting cell. Maksudnya bila macrophage ni dia dah engulf, okay, dia dah engulf pentogen tu, and so ini pentogen, okay, dia dah engulf. So inside this macrophage, okay, so dia akan degrade. Okay, it will degrade the pentogen into fragments. Okay, into fragments, we call it as antigen. So at the surface of this macrophage, it will present the antigen to another cell. We call it as helper T cell. Okay, helper T cell. Okay, so this helper T cell, once it binds to the macrophage that presents the antigen, it will activate, okay, either cytotoxic T cell ataupun B cell. Okay, so this will activate the humoral immune response. This will activate the cell-mediated immune response. Sama juga dendritic cell ni. Okay, so dia akan terlibat dalam mengaktifkan, okay, it activates the third line defense, okay, to protect your body against invasion of certain specific pathogen based on the antigen, okay, that are present from that pathogen. Okay, sebab apa? Nanti ada lymphocyte that has receptors specific to that antigen being presented by the APC, okay, antigen presenting cell, okay, which is the macrophage and also the dendritic cell. All right. Okay, so macrophage or macrophage. Okay, so macrophage, they are large, irregular shape of phagocytes known as big eaters. So another term for macrophage is big eaters. So developed from monocyte in the blood. Okay, so monocyte, kalau kita belajar, kita buat lab hari tu, dia punya nucleus dia bentuk kidney shape macam tu sikit. Okay. Right. Kalau lymphocyte, nucleus dia, it covers most of its cell. Right. Okay, so developed from monocyte in the blood, so patrol the body's tissue, engulfing damaged cell and also foreign matter. So other resides permanently in organs and tissues. Example, the lungs air sac, which is the alveolus lah, okay, the alveoli, air sac ni, contain large number of macrophage to destroy foreign matters that enters from the air that you inhale. Okay, so the air that you inhale is not 100% clean. It has other contaminants. So if these contaminants that contains, for example, virus or other pathogen manage to enter into your lung, so the defense mechanism is that inside the lungs will have macrophage to destroy, to engulf that pathogen. Okay. So this is macrophage and this is dendritic cells. Okay. As for the dendritic cell, okay, ingat yang ini pun macrophage ni akan jadi antigen presenting cell. Same goes to dendritic cell. Okay. So dendritic cells roam in tissues that are in contact with the environment. So it migrate to the lymph nodes, okay, after encountered the pathogens. So in the lymph nodes, dendritic cells will interact with immune cells to stimulate adaptive immunity. So adaptive immunity is the third line defense. Okay, third line defense, or we can also call it as the acquired immunity. Okay, acquired immunity. So this dendritic cell, when it engulfs the pathogen, it destroys the pathogen into fragments, and then these fragments, we call it as the antigen, will be presented at the surface of the dendritic cells. Okay, so it daripada dalam dia dah pecah-pecahkan kan, jadi fragments. So nanti dia akan keluarkan, okay, at the surface of its cell. Okay, ini asalnya dendritic cell. So bila dia present at the surface, dia akan jadi antigen presenting cell. Okay, so this antigen presenting cell will attract helper T cell. Okay, it attracts the helper T cell so that the helper T cell will activate cytotoxic T cell in the cell mediated immune response. Okay, cell mediated immune response. Okay, so next is the neutrophil. It is the most abundant leukocytes. Okay, so leukocyte kita ada 4. Kita ada neutrophil, kita ada eosinophil, kita ada basophil. Tapi the one that has the ability to engulf is these two, neutrophil and eosinophil. So when you are sick, okay, when contohnya bila dalam lab kan, tu ada microscope slide of blood cells. Okay, so if you were to take your blood sample, okay, and another person who is sick, okay, were to take his blood sample, and then you observe these two blood sample under the microscope, you will find that the blood samples from the person that is sick, dia demam ke apa, that person that are sick will have dia punya neutrophil dia lagi banyak. Okay, lagi banyak berbanding orang yang sihat tu. Okay. So however, neutrophil is the most abundant leukocytes. Okay. So it circulates in the blood, attracted to injured sites by signals from infected tissues. So this one nanti kita akan tengok dalam inflammatory response. Okay. And then phagocytosis, bacteria before inactivated. So it will engulf the bacteria. Okay. Next is eosinophil. So eosinophil can be found in mucous membrane that lines the digestive. Sekejap ya. Saya terdisconnect ya. Tunggu dia masuk balik. Saya guna 2 device, satu laptop, satu desktop. Desktop saya untuk record, okay. Laptop saya untuk saya conteng-conteng. So laptop saya yang terdisconnect, sekejap ya. So sementara nak tunggu saya connected balik into the Google Meet, do you have any question? Tak ada ya soalan? Ke semua tengah buka buku CTU untuk study petang ni?
Tak, madam.
Alright. Okay. Alright, okay. So, jap. Tak present pula. Okay, so continue again on the eosinophil. Okay. So eosinophil can be found in mucous membranes that line the digestive, respiratory, and also urinary tracts. So it defend against multicellular invaders. Okay, so position themselves against the parasite body and destroy the parasite by secreting destructive enzyme. So that's why tadi kita tengok dia tulis kat sini eosinophil limited roles in phagocytosis. Dia boleh engulf pentogen tapi dia juga akan secrete destructive enzyme that will kill multicellular parasite. Okay. Okay, so the second type of uh, internal defense or second line defense, internal defense, okay, is antimicrobial proteins. Okay, so peptides. So this one for antimicrobial proteins, it involves peptides and protein that kill, prevent or inhibit the growth or reproduction of pathogen. So the first one is interferon. Okay, so basically the function of interferon is to prevent a cell-to-cell spread of virus. Okay, so the virus can infect your body cell. So once your body cell is infected by the virus, so that infected body cell will produce this molecule interferon. So this interferon will find a healthy cell and dia macam warn that healthy cell so that this healthy cell produce another molecule. We call it as the uh, another molecule. We call it as the this one antiviral protein. Okay, so it produces antiviral protein. Okay, antiviral protein. So this antiviral protein will protect the healthy cell from being infected by the virus. Okay, so this one complement system. This complement ini by the host cell. Okay, yeah, interferon antiviral protein is by the host cell. It produces by the host cell. But as for the complement system, it is also the protein within the host cell. But the function of this complement system is to attack the pathogen. Okay, it is to attack the pathogen through a series of mechanisms that we are going to look at after this. Okay, so the first one, interferon, the first type of antimicrobial protein is to inhibit viral reproduction. Okay, so secreted by virus infected body cell. Okay, so this is the host cell, sel badan awak. Okay, that is infected by the virus. Okay, maksudnya sel badan awak tu kan ada virus tu. Ada virus tu. Okay. So, secreted by virus-infected body cell, stimulate in uninfected cell. So this is the healthy cell that is not infected by the virus. Okay, stimulate uninfected cells to produce antiviral protein. So this is secreted by the uninfected cell or the healthy cell. So when the uninfected body cells produces this protein, antiviral protein, it blocks the entrance of the virus inside that healthy cell so that the virus cannot reproduce inside the healthy the healthy cell or the uninfected cell. Okay, so this will help control viral infections such as cold and also influenza. Okay, by limiting cell to cell spread of the virus. So dalam awak punya persekitaran ni akan ada banyak pathogen. There are many different types of viruses in your surrounding. Okay. So when you are exposed to these viruses, so this is one of the mechanism of how your body protects you from being infected, okay, of infected by virus, okay. So by the production of interferons by infected body cell, and later by antiviral protein by the healthy cell, okay. So this prevent, okay, cell to cell spread of viruses. So other type is interfering viruses helping activate macrophages. This enhance their phagocytosis. Okay, so um, the presence of the virus will also activate a macrophage to enhance the macrophage ability to engulf that pathogen through phagocytosis. So as you can see from this diagram, so this is the host cell 1. Okay, so this is the infected cell. Okay, infected cell. So as you can see, so this is the virus outside of the cell. So virus ni, okay, sebenarnya dia akan inject dia punya, dalam virus dia tak ada DNA Virus tak ada DNA. Virus ni dia ada RNA je. Okay, RNA. So this RNA, okay, will activate a gene inside the host cell DNA, okay, which activate this gene, interferon gene. So interferon gene is activated, and then from the activation of this gene, okay, causes gene expression to occur. Okay, gene expression to occur apa? Dia melibatkan apa kita dah belajar dulu? Transcription and translation. So from here, DNA to mRNA is transcription. Okay, transcription. So from DNA to mRNA is transcription. So the mRNA will enter into the cytoplasm and produces this interferon molecule through the process of translation. Okay, so this involve gene expression lah, kan? So the presence of virus in the infected cell will activate interferon gene, so that the cell produces interferon molecule. So this interferon molecule will exit the infected cell and finds an uninfected cell. Okay, so this uninfected cell, okay, dia virus tu tak sempat nak masuk lagi, kan? So this uninfected cell is the target for the interferon. So this interferon enters into the uninfected cell and activates, okay, antiviral genes that produces antiviral protein. Okay, it activates gene that produces antiviral protein. So dia akan again stimulate or activated gene expression. So dia akan hasilkan mRNA through transcription, through translation it produces antiviral protein. So this antiviral protein will block the viral reproduction inside the uninfected cell. So this will prevent, okay, cell to cell spread of the virus. Okay, cell to cell spread of the virus. Okay. So this is a video. Okay, nak tengok ke? Kejap. Ini media satu tadi.
against viral infection. Here, a virus binds to a
Sekejap.
target.
Material into the cytoplasm. When the foreign genetic material enters the cell and starts to synthesize viral products, the cell is stimulated to make alpha and beta interferons. These interferons can signal to neighboring cells to directly inhibit viral metabolism. They do this by binding to interferon receptors, which signal the synthesis of antiviral proteins. The antiviral proteins can switch the cell into a virus-resistant state if it should become infected. Interferon gamma, produced by the activated T cells, also induces synthesis of MHC class I molecules, which make the cell more readily recognized by cytotoxic T cells. When a virus infects the activated neighboring cell, the antiviral proteins detect foreign genetic material, becoming activated, and block viral replication by breaking down the mRNA and stopping protein synthesis. These proteins put the cell into stasis, which limits further replication of the virus and
Okay. So, video dia stop halfway je macam tu lah. Tapi basically, this one is to explain on the function of interferons and also antiviral protein, okay, produced by the host cell. Okay. Alright. So next. Okay. So the second type of antimicrobial protein is the complement system. Okay, so complement system is actually a group of approximately 30 proteins in the blood plasma. Okay, 30 different proteins in the blood plasma. So these proteins will circulate in an inactive form. So it circulate in your body fluid in the inactive form. Okay, and they are activated by substances on the surface of the pentogen. So when this complement system or the proteins encounters a pentogen, it will be activated. Okay. Okay, so the activation triggers the generation of membrane attack complex. Okay, tadi kita tengok istilah apa yang tadi tu? Cellular counter attack. Cellular counter attack is phagocytic cell. So jangan confuse. Cellular counter attack, it involve phagocytes. This one, okay, is membrane attack complex. Okay, so the function of this many proteins or the complement system is to attack the membrane of the pentogen. So this one it is referring to the membrane of the membrane of the pentogen. So the protein will attack the membrane of the pentogen. That's why it called here as membrane attack complex. Okay, different many different proteins that attacks the membrane of the pentogen. So the complex insert themselves into the plasma membrane of the pentogen and forms a lethal hole or pore. Kenapa lethal? Dia akan menyebabkan plasma membrane of the virus tu berlubang-lubang. Okay. Macam tu berlubang-lubang. So once there are many pores produced at the plasma membrane of the bacteria, so water, ions, okay, can enter into the bacteria or the virus and causes the bacteria or virus or the pathogen to burst. Okay, burst, lyse, and then destroyed lah. So that's why we call it as the lethal hole. Lethal, berbahaya. Okay, bukan little lethal. Okay, so this is the complement protein. Okay, the complement system. Okay, so it consists of a series of proteins. Okay, so this protein is inserted into the plasma membrane of the bacteria. So this is the plasma membrane of the pathogen lah. Okay, pathogen. Okay, so membrane of the invading microbes. So this complement system or the protein will be inserted into the plasma membrane of the pathogen and forms pores. Okay, pores or little pores. Okay. So extracellular fluid enters into the pathogen through the pores, causing the pathogen to swell and then burst. So other complement proteins coat the surface of the pathogen to enhance phagocytosis of that pathogen by the macrophage. Okay, so another function of the complement system, apart from the cause the cell, the pathogen cell to swell and then burst, it serves also as macam recognition molecule, so that once it binds to the pathogen, it will trigger macrophage to come to that pathogen and causes the pathogen to engulf. And causes the macrophage to engulf the pathogen. So saya start dari semula. The second function of the complement system is to coat the surface of the pathogen, and it attracts macrophage to engulf the pathogen through phagocytosis. Okay, so this is complement system is the second type of antimicrobial protein. Okay, so the third of the second line defense is the natural killer cell. Okay, second line defense. Okay, so the natural killer cell or the NK cell. So they are large granular lymphocytes, okay, originate from the bone marrow, destroys virus infected body cells. Okay, the host cell that is infected by the virus will be destroyed by the NK. Okay, so the NK will also destroys tumor cells by both innate and antibody mediated processes. Okay, sebab apa dia melibatkan antibody mediated processes? Because once the NK ni dia destroy infected host cell. Okay, so they destroy. So inside this host cell dia akan ada that pathogen lah, kan? So bila host cell is destroyed, so this exposes the virus. So once this virus is exposed, akan datang antibody. Okay, antibody will bind to that virus or the pathogen. So antibody ni dia ada cara dia bertindak, ada beberapa cara. Kan, satu, dia akan cause the pathogen to be inactivated. Okay, second, it causes the antibody causes the virus to be clumped together, and then third, dia akan cause the virus or the pathogen to be engulfed by macrophage. So sebab itulah kat sini dia melibatkan both innate and antibody mediated processes. Innate is the NK. Okay, NK that destroys the host cell. Antibody sebab apa? Bila host cell is destroyed, it exposes the virus to the antibody circulating in the body fluid. Okay, so antibody is produced by the B cell. B cell is this antibody mediated processes, ataupun another term for antibody mediated processes is humeral. Okay, humeral. immune response. Okay, so dia saling interconnected lah. So second line defense dia akan bekerja sama juga dengan third line defense. Okay. Okay, so next, how does the NK destroys the infected body cell? It releases two kind of chemical molecule. The first one is perforin. Okay, so NK will release perforin, which is a type of protein that will form pores. Okay, at the membrane of the target cell. Macam ni lah, lebih kurang macam ni. Okay, so first it secrete perforin that forms, which is protein that will be inserted into the membrane of the pentogen and causes the formation of pores. So nama pun perforin kan? Okay, proteins that will be inserted into the membrane of the pentogen to form pores. Second, it releases this second molecule, which is granzyme. Okay, natural killer cell will secrete these two molecule. First is perforin, second is granzyme. Okay, so granzyme, nama dia pun awak boleh tahulah. So it is a protein or an enzyme, okay, protein that enters into the cell and activate proteins inside the cell, which induces apoptosis. Okay, apoptosis adalah it induces the whole cell to be destroyed. That is apoptosis. Kita tengok dia punya definition. Saya lupa dia punya istilah dia. A form of program cell death, okay, or cellular suicide. So it causes the host cell, okay, to undergo program cell death, okay, ataupun cellular suicide. Okay. Okay. So that is how the natural killer cells destroy the infected body cell or the infected host cell. Okay. Release two molecules, perforin and also granzyme. So ingat natural killer cell ni dalam second line defense, innate immunity. You have another type of cell which is we call it as the cytotoxic T cell. Cytotoxic T cell mechanism dia sama macam natural killer cell. It also releases perforin, it also releases granzyme, but for cytotoxic T cell it is in the third line defense. Okay, the mechanism posed by the cytotoxic T cell is more specific. It destroys specific virus. Okay, it destroys infected cells that is infected by specific virus. Tapi kalau yang ni more generalized way, in a generalized way, macam tu. Dia tak specific. Okay, innate. Okay, so this is a diagram to show you how the natural killer cell will destroy the host cell. So this is the host cell, the target cell, infected cell. And so this is the natural killer cell. Awak tengok natural killer cell tadi dia adalah lymphocyte. Lymphocyte ni, mana lymphocyte tengok dia punya nucleus dia, it covers most of the cell. Yeah, so itu adalah lymphocyte. And then as you can see from this diagram, natural killer cell will approach the infected body cell. It releases first is perforin, kan, perforin yang biru-biru ni, kan. So it releases perforin, so it forms pores, kan, at the membrane of the infected host cell. Okay, so once spores are produced, then it insert the second molecule, which is granzyme. So this granzyme will induce apoptosis, okay, which is program cell death. Okay, so the apoptotic cell is broken down into vesicles, and then these vesicles from the infected body cell, from the destroyed infected body cell, will be engulfed, okay, by macrophage. Okay, same. So they akan engulf the debris of the infected body cell and also the exposed virus. Okay, the exposed pathogen. Okay. Okay, so this the fourth component of the second line defense. Okay, still on the second line defense, is the inflammatory response. Okay, so again, because it is second line defense, innate immunity, so it is non-specific. Okay, non-specific response. Maksudnya dia tak target mana-mana specific pathogen. Mana yang ada injured tissue, inflammatory response will take place. Okay, so non-specific response to infected tissue or injured tissue. Okay, to infection or injured tissue. Okay, so minor injury or infection causes local inflammatory response, but more severe injury or infection may lead to systemic inflammatory response. Awak demam tu, we call it as systemic inflammatory response. Systemic inflammatory response, it means that the fever occurs throughout your body. Tak adalah tangan awak je yang panas kan, kepala awak je yang panas kan. Seluruh badan awak akan panas. Okay, seluruh badan awak panas. That one we call it as the systemic inflammatory response. Daripada nama tu, inflammatory, inflame. Kawasan yang kena infection tu akan berasa panas. Sebab apa dia berasa panas? Akan berlaku vasodilation. Okay, so when vasodilation occurs, blood vessels, its diameter will dilate. It brings more blood to the infected tissue or area. This will bring more metabolic heat. Okay, more metabolic heat. So that's why area yang kena infected tu, kalau awak ada terluka ke, awak ada infection lah at certain area, so that area yang ada merah-merah tu akan berasa panas. Kalau demam lagi lah rasa panas kan. So that's why the name, we call it as the inflammatory response, due to vasodilation of blood vessels that brings more blood and brings more metabolic heat. Okay. Okay, so local inflammatory response. Satu kawasan tisu tu yang kena infected. Okay, local. So characterized by redness, warm, redness, warm, swelling. Swelling is edema kan, accumulation of tissue fluid at that infected area, and also there's pain. Okay, akan rasa sakit jugalah kat situ kan. So this involve, kenapa akan rasa sakit? Sebab bila berlaku edema ni, swelling, okay, it presses against the nerve tissues, okay, nerve cells. So that's why akan rasa pain. Okay. So mast cell at infected or injured site releases histamine. So mast cell ni adalah sel pada tisu yang infected tu. Pada tisu yang infected tu akan ada mast cell. So this mast cell akan store vesicles that contain histamine. Okay, mast cell will contain vesicles that contains histamine. So when you are infected, okay, when a tissue is infected, so this mast cell releases histamine that have two functions. One, it causes vasodilation, okay, of nearby blood vessels. Blood vessels dilate lah, kan? Dilate. Okay, dilate, bring more blood, okay, to the injured tissue. Okay. So another effects of histamine is that it increases the permeability of nearby capillaries. Okay. So capillaries dia duduk kat mana? Tissue kan. So it causes the capillaries to increase its permeability. Maksudnya apa? Blood capillaries ni akan ada sel-sel darah kan. Dalam sel-sel tu akan ada white blood cell, especially neutrophil. Okay, so by increasing the permeability of the capillaries, neutrophil that was inside the capillaries will come out of that capillaries into the interstitial fluid surrounding the injured tissue. So neutrophil akan keluar. So akan keluar daripada blood capillaries tu. Okay, into the interstitial fluid surrounding the infected tissue. So this is so that if there's bacteria or pathogen in that area, so the function of the neutrophil, for example, is to engulf any pathogen that is inside the tissue, inside the infected tissue. Okay, so if you look at this diagram, so nampak. So first, okay, this is the mast cell. Mast cell releases histamine. So the function of histamine is one, to cause vasodilation. Vasodilation, nampak daripada kecil dia jadi besar. Okay, it brings more blood. And then second, it causes or increases permeability of the blood capillaries so that neutrophil, for example, can come out of that capillaries into the tissue fluid or interstitial fluid or tissue fluid of the infected area here. Okay. So, vasodilation of local blood vessels kan disebabkan oleh histamine tu tadi. Okay, histamine. Vasodilation of local blood vessels increases the blood flow to the injury site. Okay, saya dah mention tadi. So this causes the area to become red. So bila awak luka, kawasan tu akan jadi merah. Okay, sebab due to vasodilation brings more blood to the infected area. Okay, so second, warm. Okay, because more metabolic heat is carried by the blood. Okay, so that's why nama dia inflammatory response. Okay, dia akan menyebabkan kawasan tu merah dan sakit kat situ. Okay, dan rasa panas. Okay, so local inflammatory response, tadi yang first effects of histamine kan, vasodilation. Second effects of histamine, okay, it increases capillary permeability that leads to the release of fluid, blood clotting elements and phagocytes such as monocyte and also neutrophil to the injured site. So permeability of the blood capillaries increase so that fluid from the blood enters into the tissue fluid, and also when fluid from the blood enters into the interstitial fluid, it also brings out clotting element, okay, and also white blood cells, phagocytes, monocyte and also neutrophil to that injured site. Okay, so this causes edema. Edema ni lah tissue swelling disebabkan apa? Fluid yang keluar daripada blood capillaries tu tadi, tissue swelling, kawasan tu bengkak. Okay, and then pain. Okay, so pain because of the pressure on nerve endings on the region due to edema. Okay, so kawasan tu akan berasa sakit lah sebab apa? Ada pressure due to edema. Okay. Okay, so still on local inflammatory response. So blood clotting elements. So the function of blood clotting elements yang keluar daripada the blood capillaries tu due to increased permeability of the blood capillaries. Okay, so the blood clotting elements will wall off inflamed area to prevent or delay the pathogen or toxic from spreading into surrounding tissues. So clotting elements yang keluar kat infected area tu fungsi dia adalah untuk block the spread of pathogen from spreading into other areas. Okay. And then neutrophil, to carry out phagocytosis, secrete signaling molecules that attract monocyte that will differentiate into macrophage to carry out phagocytosis. Kan? So yang disebabkan oleh apa? Increased permeability, neutrophil keluar daripada blood capillaries into infected tissue. Okay, so neutrophil will engulf any pathogen that enters into the infected tissues, and also they akan release signaling molecules that attract monocyte. Monocyte ni bila dia mature dia jadi macrophage. Macrophage akan engulf pathogen, okay, through phagocytosis. Okay, so activated neutrophils and macrophage releases this molecule which is cytokine. Okay, so cytokine will increase blood flow to the injury site, which also leads to redness and warmth. Okay, so ada dua molekul lah ni tadi. Satu histamine. Okay, histamine by mast cell. Okay, mast cell. So histamine tadi dia buat apa? Vasodilation and increase permeability of blood capillaries. Okay. Second, bila increase permeability, apa yang akan keluar? Neutrophil, monocyte. Monocyte akan mature jadi macrophage. Okay. So, bila dah banyak-banyak neutrophil keluar into injured tissue, so dia akan release these two cell, macrophage and also neutrophil akan release cytokine. Okay. These two akan release cytokines. Neutrophil and macrophage releases cytokine so that they akan cause more redness and more warmth due to more blood flow to that injured area. Okay, so phagocytosis by the phagocytic cell leads to the formation of pus. Okay, that is a mixture of dead pentogen, tissue cells and also phagocytic cell. So, inilah kan kawasan yang luka tu tadi tu kan ada bengkak, ada combination of white blood cells, pentogen, all the reaction inside it, kan all the fluid we call it as the pus. Okay, pus that is a mixture of dead pentogen, tissue cells and also phagocytic cell. Okay, so this is the inflammatory response. If you look at the last diagram here, number 3, neutrophil digest pentogen and cell debris and the tissue heals. Kalau tak silap saya yang saya pernah baca dulu, okay, salah satu dia punya benefit of that area being tadi dia cakap apa? Painful kan? Akan rasa pain tadi tu. Mana tadi? Ni kan? Pain. Okay. Sebab apa? Sebab kat situ dia bila terasa sakit kat situ, kawasan tu awak akan tak ganggu dia, tak pegang, tak kacau semua. So this will allow the area to heal. Okay. So itu sebab akan rasa painful. Okay, so that area will be left alone so that it gives time for the tissue to heal. Okay, tadi local kan? Local, satu kawasan tissue tu is infected. Okay, local. If your body, the whole of your body is infected, okay, especially there's a virus inside your body fluid, we call it as systemic inflammatory response. Okay. So it says here, toxin released by pentogen and phagogen produced by macrophage lead to the increased body temperature greater than 37.2 or 68.6 Fahrenheit. Okay, causing fever. So kalau demam ni kan, bila selalunya suhu badan awak nak cecah 37.2 ni akan terus rasa nak demam lah. Selalunya kalau macam kita ambil termometer kan, sekarang ni ambil termometer, selalunya termometer awak akan read 36 point something. Tapi kalau dia dah start nak masuk 37.2, 37.3 tu maksudnya nak dekat demam dah. 37.5 tu maksudnya memang demam. Okay. So ini 37.2 will cause fever lah. Okay. Okay, so the benefit of fever is to promote phagocytosis by phagocytic cell. So the increased temperature, the increased body temperature will promote phagocytosis by the phagocyte, okay, to engulf the pentogen. Second, it inhibits the growth of some pentogen by stimulating the liver and spleen to store iron so that blood iron level is reduced. Okay, so another benefit of fever is that dia akan limit the growth of pentogen. Macam mana cara dia limit growth of pentogen? Pentogen ni, okay, sel pentogen ni dia perlukan iron. Okay, iron, ferrum lah. Okay, so cell of the pentogen requires iron. If the host cell, if the host stores the iron, okay, in the liver, so maksudnya dalam badan orang tu dah kekurangan iron. So mana bakteria tu nak dapatkan iron? So dia tak boleh nak hidup dah lah, faham tak? So another benefit of fever. It causes your body to store iron in the liver, so there's no iron for the bacteria to depend on, because the iron is needed by the pathogen for its metabolism. Okay, to so that the pathogen can continue reproducing. So if there is no source of iron, the iron is stored in the liver due to the high temperature of the body. So the pathogen reproduction and growth will be prevented. Okay, macam tu. And then the next one is it accelerates tissue repair, okay, by speeding up chemical reactions. So these are the three benefits of fever. Okay, so sebenarnya fever tu adalah cara badan awak untuk recover lah against infection. Okay. Okay, so finish with the second line defense. So do you have any questions? Ada soalan?
Tak ada, madam.
Tak ada. Okay. So if there's no question, so I will proceed to the third line defense. Okay, third line defense component of the third line defense. Okay, ini tajuk separate third line defense apa dia. Okay. And then components of the third line defense. Okay. Okay, so here another term for third line defense is adaptive immune response or acquired immunity lah. Okay, so it is a specific defense system. Bila kita dah masuk kepada third line ni, it is specific. Okay, due to the presence of receptor on the lymphocyte. On the lymphocytes. Okay, so this is to detect specific pathogen. Okay, so it is a specific defense system which also known as the third line defense, detects very specific pathogen using a small group of receptor proteins. Okay, so the response is slower. Kalau innate tadi, first and second line, the response is rapid. But for the third line defense, the response is slower. Itu perbezaan dia. Okay, so third line defense is activated after innate immunity takes place. So nampak tadi kena lalu dulu first line defense, second line defense, baru third line defense will take place. Okay, so found in the vertebrates, two type you have the antibody mediated immunity or known as the humoral immune response. So humoral immune response, why? Because it involves body fluids. Okay, body fluids such as blood, interstitial fluid and lymph. Okay, kenapa juga kita panggil dia sebagai antibody? Because this one, the humoral immune response will activate B cell. Okay, so B cell, when it is activated, it will produce and secretes antibody. Okay, antibody against that specific pathogen. Okay, so another type is cell-mediated immunity. So this one, it involves the cytotoxic T cell. Okay, cytotoxic T cell. So both of these cells, okay, they are lymphocyte. Okay, they are lymphocytes. Okay, so adaptive immune response. So we have two, which is the first one is antibody mediated immunity or humoral immune response. Second, you have the cell mediated immunity. Cell mediated, saya lupa nak cakap tadi, it attacks infected body cell. Okay, infected body cell. Yang ni body fluid kan? So yang ini dia akan attack. Sinilah tak muat. Infected body cells. Okay. So for the first one, antibody mediated immunity or humoral immunity or response involve the activation and clonal selection of B cells which secretes antibody. Okay. So clonal selection ni maksudnya bila B cell tu dah diaktifkan, activated B cell, okay, when it is activated, it undergoes clonal selection. So clonal selection maksudnya akan ada dua koloni yang terhasil. The first one is activated B cell, we call it as the plasma cell. Plasma cell or activated B cell. The other one is we call it as the memory B cell lah. Okay. Memory B cell will be stored in the lymph nodes. Okay. So same goes to the cell mediated immune response. Before we go to the cell mediated, saya lupa nak bagi tahu, this plasma cell, okay, will produce and secretes antibody. Antibody that will circulate in blood, interstitial fluid, and also lymph. Okay, circulate so that it will find that specific pathogen in either of these three body fluids. Okay. As for the cell-mediated immunity, involve the activation and clonal selection. Nampak bila dia cakap activation and clonal selection, mesti menghasilkan activated cell and also memory cell. Okay, memory simpan, activated cell untuk bertindak pada masa infection tu. Okay, so memory untuk simpan untuk future infection by the same pathogen. So involve activation and clonal selection of T cytotoxic cell that recognize and destroy the pathogen. Okay, specifically it destroys infected body cell by that specific pathogen. So once the cell is destroyed, the pathogen is also will be destroyed. Okay. So major characteristics of adaptive immune response. One is rapid response to second exposure of the same antigen. Tadi kalau first exposure, the response is slow. Tapi sebab due to that first exposure akan terhasil memory kan, memory cell, memory B cell ataupun memory T cell. So this memory B or T cell that is stored in the lymph nodes will be stored for many years, for decades. Okay, so if your body is infected again by the same antigen of the same pathogen, so your body will be automatically respond to that invasion of the same pathogen. So the response is rapid. Maksudnya tadi tak payahlah nak kena engulf by macrophage dulu, lepas macrophage tak payah nak pergi cari helper T cell untuk activate helper T cell. So tak payahlah nak cari B ataupun T cell nak activate, undergo clonal selection. So cerita dia panjang. Okay. Itu kalau first exposure. Tapi second exposure, bila dah terhasil je memory, when you are infected by the same virus again, your body will be automatically defend against that virus or against that pathogen due to the presence of the memory cell. So perbezaan antara first exposure and second exposure is that first exposure response is slow. Second exposure or subsequent exposure, the response is much more rapid due to the presence of the memory cell. Okay. Second, detect antigen never before encountered. So that is dia punya kelebihan dia lah kan. Dalam badan awak ni, there are millions and millions of different type of lymphocyte. Okay, you have many different lymphocyte. Awak ada contohnya, contohlah saya bagi kan, B cell 1, B cell 2, B cell 3. Okay, so this B cell on its cell surface ada receptor. Okay, there are thousands of receptor on the surface of the B cell. Okay, so this receptor dia akan ada binding site for the pathogen. Tapi untuk setiap B cell yang sama ni, dia punya receptor dia sama je. So maksudnya dia akan detect the same type of pathogen. Sorry, the same type of antigen. So in this case, antigen yang dia akan B cell yang pertama akan detect antigen yang bentuk bulat contohnya. Okay. Tapi kalau badan awak ni diserang oleh second type of pathogen. Okay. Contohlah. So yang berbentuk 3 segi ni. So B cell 2 yang akan diaktifkan. Tapi kalau contohnya yang lain, antigen yang bentuk 4 segi pula serang awak kat sini. Kan? So antigen yang bentuk 4 segi akan aktifkan B cell yang ketiga. So ini baru 3 B cell. Dalam badan awak ada banyak beribu-ribu, berjuta-juta kan, berjuta-juta white lymphocyte that each one will have different receptor. Okay, each B cell in your body will have different receptors. So this receptor will detect different shape of antigen. Okay, so so dia akan increase. Okay, it increases the possibility of the B cell to detect any pathogen that enters into your body. So once the B cell has detected the pathogen, it triggers the activation of your immune cells against that pathogen. So that's why dia cakap nombor 2 ni, detect antigen never before encountered due to the presence of many B cells that each one has different shape of binding, antigen binding site. Okay. The third one, able to distinguish self and non-self. So your immune cells, okay, as I mentioned, it only attacks non-self antigen, which is foreign pathogen that enters into your body. Your immune cell cannot attack self antigen. Self antigen maksudnya antigen daripada host cell, badan awak sendiri. So your immune cell cannot attack your own body. Okay, it cannot attack self antigen. If your immune system attacks your own body cell, it could lead to autoimmune disease. Okay, so hanya your immune system only detects and attacks non-self antigen. Okay, number 4, cell proliferation. Okay, increases the number of B and T cells specific for that antigen. So this is antigen of the foreign molecule, antigen of the pathogen. Okay, antigen of the pathogen, kan? So apa maksud cell proliferation tu? So let's say lah, dalam satu keadaan tu badan awak kena attack by pathogen yang ada antigen bentuk 4 segi ni. Okay, so this B cell yang ketiga ni will be activated. Once it is activated, it will proliferate. Apa maksud proliferate? It will divide into many copies, many cells of B cell tree ni. It will proliferate. Okay, produce many B cells with the same receptor. Okay, with the same receptor against that specific pathogen. Tu contohlah. Okay, so proliferate into many cell that later will undergo clonal selection. Clonal selection ni lah, nak select yang mana? Yang A ke B ke? Sorry, B1 ke B2 ke B3. So in this case is B3. So clonal selection. So dia akan jadi nanti 2 clone, 1 memory. Okay, 1 lagi activated. Okay, ini saya bagi contoh untuk B. Sama juga mekanisme untuk T cell. Okay. Okay, so cell says here, cells involved in adaptive immune responses. So there are 2 key cell types, which is lymphocyte and antigen presenting cells. Okay, antigen presenting cell ni dia ada 3 jenis actually. Okay, 1 macrophage, 2 dendritic cell, 3 B cell pun boleh jadi. Okay, B cell. Okay, B cell pun boleh jadi antigen presenting cell ataupun APC. Lymphocyte kat sini kita ada B cell, kita ada kita ada T cell lah ataupun cytotoxic T cell. T cell ni, okay, T cell ni ada dua jenis. Okay, kita ada cytotoxic. Cytotoxic T cell. Lagi satu kita ada helper T cell. Okay, ingat lymphocyte. Okay, macam itulah. Okay, so lymphocyte include T lymphocyte, T cells and B cell, B lymphocyte. Okay, so T cells and B cell recognizes antigen. Sebab apa? Yang macam saya draw tadi, kalau B cell, dia punya structure dia, antigen dia bentuk bentuk Y shape. Macam tu. Bukan macam ni, macam ni, Y shape. So each one ni ada antigen binding site. Antigen binding site. So each of these antigen binding site will bind to the same shape of antigen of the pentogen. Okay, ni B cell punya receptor. Kalau T cell, dia satu macam tu je. Ini antigen binding site, satu je bentuk dia. Satu dia bukan Y shape. Macam tu. Okay. B cell dia punya antigen, dia punya receptor bentuk Y shape. Okay. So all lymphocyte develop from stem cell in the bone marrow. So semua ni terhasil dekat bone marrow. Okay. But B cell completes its development in the bone marrow, but T cell matures in the thymus. Okay. Kesemua lymphocyte ni terhasil dekat bone marrow, tapi untuk menghasilkan B cell, lymphocyte tu akan maintain dalam bone marrow and mature in the bone marrow. But the one that will become T cell will go to the thymus and mature in the thymus. Thymus tu gland kan? Gland yang ada dekat atas jantung. Okay? Thymus gland. So roam in the tissues and organ of the lymphatic system to induce immune responses. So dia akan circulate, okay, circulate in body tissues and organs of the lymphatic system to induce immune responses. So bind to antigen and undergo clonal selection to produce clone of cell. Okay, clone of cells. Clone tu tadi saya cakap apa? Sama ada activated, okay, clonal selection lah, activated ataupun memory. Okay. Okay, so two types of lymphocyte here. Perbezaan dia antara B cell dengan T cell. So B cell matures in the bone marrow. T cell mature in the thymus. Okay, so B cell, once it is activated, the activated B cell, we call it as the plasma cell. Okay, so the plasma cell will produce and secrete antibodies. The antibodies secreted by the B cell will be the same shape, okay, the same function as the receptor that are found on the B cell surface tadi. Cell surface tu tadi kan. So shape antibody yang terhasil daripada B cell akan sama dengan receptor yang ada pada dia punya plasma membrane. Okay, so this for B cell it activates or mount or involve in the antibody mediated immune response ataupun / kat situ humoral immune response. Okay, so for T cell, so tadi apa, all the lymphocyte are produced in the bone marrow, tapi yang akan jadi B cell dia akan mature and maintains in the bone marrow. Tapi kalau untuk yang T cell dia akan mature in the thymus gland, in the thymus. Okay, so directly attack the cells infected by the pathogens. So because it attack infected body cells with that specific pathogen, it involved in the cell mediated immune response. Okay, so two types, you have the T cytotoxic cell, okay, T cytotoxic cells, okay, T cells. Kena letak cytotoxic ni sebab kita ada dua tadi, helper T cell dengan cytotoxic T cells, okay, that destroy cells infected with the pathogen. Second, you have ada dua jenis T cytotoxic cell, and the second one is helper T cell. Saya dah terbiasa sebenarnya. Saya sebut dia helper T cell. Kat sini tulis T helper cell. Sama je lah. Okay. T helper cell dengan T cytotoxic cells. So kalau T helper cells, okay, that activate B cells and cytotoxic T cell by secreting cytokines. So maksudnya kat sini daripada macrophage contohnya, macrophage, dia engulf pentogen jadi APC kan. Lepas tu daripada APC dia akan APC will attract helper T cell. This helper T cell will activate both, okay, cytotoxic T cell and also B cell. B cell dalam humoral immune response, yang ini dalam cell mediated immune response. Okay, cell mediated immune response. So kena tahulah helper T cell ni sebenarnya dia diperlukan untuk activate kan B cell dengan cytotoxic T cell. Okay. So if you look at this diagram, okay, antibody mediated immunity. So kalau tengok kat sini, okay, so you have your bone marrow. Inside the bone marrow, you have the stem cell. So the stem cell matures into immature lymphocyte. It becomes immature lymphocyte. So this immature lymphocyte, ada yang keluar pergi kat thymus and mature into T cell. Okay, matures into T cell. Tapi yang maintain akan mature into B cell. So as you can see, both of these lymphocyte at its cell surface, and cell surface dia akan ada antigen receptor. So this is antigen receptor. For B cell, the antigen receptor is Y-shaped. Okay, so each one here we call it as a antigen binding site. Sama juga kat sini. Kalau B cell, satu reseptor ni akan ada dua antigen binding site. Tapi kalau kat T cell, satu antigen receptor akan ada satu sahaja antigen binding site. Okay, it binds to the antigen of the pentogen. Okay, so both of these B and T cells will be stored in the lymphatic organs. Okay, so these lymphatic organs can be lymph nodes, can be spleen, and other lymphatic organs. Tonsil kan, other lymphatic organs, you have the tonsil. Okay, so final maturation of B and T cell in the lymphatic organs. Okay, so itu perbezaan antara B cell and T cell. So sekarang ni kita tengok T cell receptor. T cell receptor short form as TCR. T cell receptor. And another one is B cell receptor. Okay, so it is a protein that is bound to the membrane of the T cell and B cell respectively. Inilah reseptor ni. Yang warna-warna purple yang saya dah bulat-bulatkan tadi. Itu kita namakan sebagai receptors. Okay. So they are bound to the membrane of the lymphocyte. So the TCR binds to the antigen fragments that is displayed or presented on the surface of the host cell by major histocompatibility complex ataupun short form as MHC. MHC ni sebenarnya dia adalah sejenis protein yang dihasilkan oleh nucleated host cell. Kita ada dua jenis MHC. MHC class 1 dengan MHC class 2. Okay, so nanti kita tengok apa perbezaan antara MHC class 1 dengan MHC class 2. Okay, so ingat T cell ni kita ada dua, helper T cell and cytotoxic T cell. Okay, so each of this one, helper T cell dengan cytotoxic T cell ni akan bind by antigen presented by either of these class of MHC. Dia spesifik. Nanti kita tengok yang mana bind kat mana. Okay, kejap lagi. So B cell receptor binds to the antigenic determinant ataupun epitope of intact antigen on pentogen or circulating freely in the body fluid. Okay, awak tengok ya istilah ni carefully. Ni saya draw Ini adalah virus. Okay. Dekat virus. Okay, virus. Virus satulah saya tunjuk. So virus ni dekat cell surface dia akan ada contohnya antigen. Okay, antigen. This is antigen. So dekat cell surface dia akan ada banyaklah antigen. Antigen of the same shape. Dekat antigen ni akan ada small structure yang lebih spesifik. So this small structure we call it as the epitope. Okay, the epitope. Okay, so for B cell receptor ataupun T cell receptor, to be specific, it binds to the epitope of the antigen of the pentogen. Faham tak? Okay, B cell receptor binds to the, saya rephrase balik lah, B cell receptor ataupun T cell receptor will bind to the epitope of antigen on the pentogen. Macam tu. Okay. So macam contohnya, okay, ini adalah B cell, satu contohnya. Dekat cell surface dia ada receptor. Receptor dia bentuk Y shape kan? Okay. Dekat receptor, dekat each of the receptor ada antigen binding site. Okay, antigen binding site. So this antigen binding site actually binds to the epitope on the pentogen, on the antigen of the pentogen. Okay, so dia bind actually to the epitope of the antigen of the pentogen. So ingat tu. Okay. So this is a clearer diagram of the T cell receptor and also B cell receptor. Ini B cell receptor lah. So as you can see, the B cell receptor is Y-shaped. Okay, Y-shaped. It has two antigen binding site. So yang ini yang akan bind kepada epitope. Okay, on the antigen of the pentogen. So kalau awak tengok kat sini, kalau untuk T cell receptor, dia ada satu je antigen binding site. Okay, so kita tengok apa yang ditulis kat sini. A B cell receptor consists of two identical heavy chains and two identical light chains linked by disulfide bridge. Okay, kalau kat sini awak tengok kalau receptor ni, dia terdiri daripada receptor ni sebenarnya dia adalah polypeptide chain. Dia adalah polypeptide. Dia adalah protein. Okay, mana? Dia adalah protein. To be specific, dia adalah polypeptide chain. Okay, polypeptide chain. Sebab tadi saya dah mention, reseptor pada B cell ni adalah sama dengan antibody yang dihasilkan oleh B cell. Awak dah belajar daripada dulu lagi. Antibody adalah protein. Okay, antibody adalah protein. So same goes to the receptor of the B cell and also T cell. Dia adalah sebenarnya polypeptide chain. Kenapa kat sini dia cakap apa? Heavy chains and also light chain. So kat sini chain dia terdiri daripada dua jenis chain. Okay, yang identical. So this is the heavy chain. So this is the light chain. Okay, sama juga. Tapi kalau T cell receptor consists of one alpha chain. So yang panjang sikit ni alpha chain, yang pendek sikit adalah beta chain. Kenapa panggil dia chain? Sebab dia adalah polypeptide chain. Okay, so both of these chains are linked together. Dia diikat oleh disulfide bridge. Okay, disulfide bridge. So apa yang CV ni? CV ni apa? CV tu dia stand for constant polypeptide chain. Yang ni variation, tak silap saya, variability, variation, variable region, tulis kat sini. Okay, constant region, variable region. Okay, V stand for variable region, C stand for constant region. Okay, so kenapa ada constant region, kenapa ada variable region? Constant region ni maksudnya sequence of amino acid at the constant region are the same for every B cell. Okay, for every B cells, the constant region of the receptor, the sequence of amino acid is the same. But for the variable region, for every B cell tu, dia punya variable region, dia punya amino acid sequence is different. Why is it different? Sebab dia akan menentukan the shape of the binding site. Okay, variable region. So variable region is the sequence of amino acid is different from one B cell to another B cell to form the antigen binding site. So sama juga. Constant region, the sequence of amino acid for the constant region is the same for every T cell, but different for every T cell because it will determine the shape of the antigen binding site. Okay. Okay, another region yang awak kena tengok kat sini adalah untuk untuk menanamkan receptor ni pada plasma membrane kan. Region ni the region that is embedded inside the plasma membrane of the lymphocyte here, this region we call it as the transmembrane region. Okay, the region that is embedded in the plasma membrane of the lymphocyte, either B cell or the T cell. Itu je lah kan. So dia akan extend sikit into the cytoplasm. Okay. So ini adalah shape of the receptor that you have to know. Pernah keluar dalam exam pasia-pasia dulu, kalau siapa yang rajin tengok pasia kan, dia pernah suruh lukis shape of the receptor, dia pernah suruh label kan. Label ni apa benda? Dia pernah tanya. So you have to know the structure of the B cell receptor, the structure of the T cell receptor. Okay, so now we are going to look at the definition. So Pentogen tadi lagi kan. So what is Pentogen? So Pentogen is an organism or virus that can cause disease if it enters into your body. Okay, so Antigen is a substance ataupun substance that elicits immune response. Dia akan trigger your immune system to respond to that pathogen. Okay, so the antigen, it belongs to the pathogen lah. Okay, so has several different antigenic determinants, which is epitope. Okay, so on its surface induce an immune response recognized by different T cell and B cell. Has several antigenic determinants. Kalau macam saya draw tadi kan, itu saya bagi contoh yang mudah lah. Okay, so ini ada virus yang kedua. Okay, so dekat virus yang kedua ni akan ada dia punya antigen. Okay, antigen ni saya lukis petak ni supaya senang nak nampak lah. Okay, so this is the antigen. Okay, on the virus or on the pathogen. So epitope dia, antigenic determinant, dekat dia punya antigen tu ada berbagai bentuk. Ada yang bulat, ada yang 4 segi lagi. Contohnya kan, ini saya nak bagi contoh mudah faham lah. Okay, ada yang 3 segi, ada yang shape lain macam ni kan. Ada whatever lah, terbalik macam ni. Ada yang macam ni. Okay, so by having antigen with different epitopes, this increases the chance of the virus to be detected by your body. So maksudnya kalau ada B cell satu yang dia punya antigen binding site dia bentuk bulat, macam tu. Okay, bentuk bulat kan, bentuk bulat. So B cell 1 ni akan bind kepada virus tu, and this one will be activated. Tapi kalau on this side ada B cell yang kedua kan, so dia ada receptor. Ni saya nak bagi awak faham mudah. Okay, antigen binding site dia yang bentuk contohnya 3 segi macam tu. So dia akan bind kepada yang ni lah kan. So B cell tu will be activated also. Okay, so itu dia punya hikmah dia lah. So by by the pathogen having many different epitopes on its cell surface, so this will increase the chance of the virus being detected by your body. Okay, so this will activate your immune response against that virus. Okay. So the binding of the antigen to the B cell receptor initiate the production of antibody from the plasma cell. So plasma cell is the activated B cell. Okay, sebab selepas undergo clonal selection tadi kan ada dua colonies, activated B cell and memory cell. Activated B cell adalah plasma cell yang menghasilkan antibodies. Okay, against this virus yang kedua ni, macam tu. Okay. So next, antigen. So antigen is a large molecules, either proteins or polysaccharides, recognized by lymphocytes, elicits a specific immune response. Okay, maybe tadilah kan, it may be the component of the pathogen, components such as toxin. So the virus or the pathogen can also release toxin kan. Tadi toxin that release that circulates in your body akan menyebabkan tadi apa, fever kan, right? Okay, so components of the pathogen such as toxin that are released into the extracellular fluid. Okay, so proteins or glycoproteins on the surface of the pathogens, food or pollen such as the surface of the surface of the transplanted organs. So antigen is whatever foreign to your body. Okay, antigen yang akan induce the activation of your immune response. Okay, whatever that is foreign to your body will trigger the activation of your immune response. So it can be the antigen on the pathogen, it can be food. So some people are allergic to food kan, seafood ke, ada orang yang allergic kepada ayam juga ke, any type of different foods that may trigger allergic reaction, contohnya adalah disebabkan oleh that foreign molecules that is labeled as antigen by your body, foreign antigen by your body to trigger an immune response against that antigen. So pollen pun sama. Okay, so any foreign substance that enter into your body that triggers your body to respond to that foreign molecule. Sama juga dengan transplanted organs. Okay, transplanted organ ada chance to be rejected juga. Sebab apa? Sebab obviously every cell akan ada antigen. Okay, so with the entrance of the antigen into your body, it will trigger your immune response. Okay, to fight against that molecule that enters into your body. Okay, so ini contohlah. Kita ada bakterium, kita ada virus. So molekul yang kat luar-luar ni considered as antigen ataupun substance yang secretes kan, yang jadi toksin kan, molecule secreted by this organism is also considered as antigen. Okay. So kita tengok last istilah ni, antibody Last, antibody also known as immunoglobulin, Ig. Okay, so dia adalah protein juga. Okay, so it is a protein secreted by B cells. Okay, so has antigen binding site that is complementary to the antigenic determinant or epitope found on antigen. Okay, so binds to an intact antigen, interferes with the pathogen activity. So tadi ni kan kita ada virus. Ada banyaklah virus dalam host ni. Contohnya virus yang ketiga. Okay. So virus yang ketiga dekat cell surface dia, okay, akan ada antigen, right? Okay. So dekat permukaan dia ada epitope. Okay. So bila adanya virus tu dan dia akan activatekan certain B cell. So the B cell will secrete antibody. Okay. So this antibody will bind to that virus. Antibody ni cara dia bertindak ada several ways. Yang one of it dia akan neutralize the virus, neutralize the pathogen. Neutralize tu maksudnya dia akan menyebabkan virus tu tak tak boleh nak bergerak lah, faham tak? Neutralization ataupun clumping ataupun agglutination. So those are different ways of how the antibody works. So bila dia bind kepada antibody, so virus ni dah tak boleh bergerak dah. So what happen next is that macrophage akan datang, okay, and then engulf that pathogen. Macrophage will come and then engulf that pathogen that is being inactivated by the antibody. Okay. So that's why dia cakap kat sini interferes with the pathogen activity. So once antibody binds to the virus, virus tu tak boleh nak buat apa dah. Okay. So ini contoh antigenic molecules. It can be on the surface of pathogen ataupun dia can be toxin, okay, released by the pathogen, ataupun dia boleh ada pada food tadi kan, ataupun dia boleh ada pada pollen. Okay. So dekat surface ini akan ada banyak jenis epitope. Ada yang bulat, ada yang 3 segi, ada yang 4 segi, whatever. So this is to increase the chance of the antigen molecule to be detected by your immune system. Okay, by the antibody produced by your body. Okay. So this is the shape of the antibody. As I said, it is the same. It has the same shape. It has the same structure as the receptor of the B cell produced by the. It has the same. It has the same shape. Okay, same shape as the receptor of the B cell produced the antibody. Kan? Kecuali antibody ni dia tak ada transmembrane region. Okay, antibody dia tak ada transmembrane region je, sebab transmembrane region adalah region yang embedded in the plasma membrane of the B cell. So itu je perbezaan dia. So antibody has the same shape as the receptor on the B cell that secretes the antibody, kecuali tak ada transmembrane region sahaja. Okay. So now kita stop dulu. Okay, dah rasa dah banyak dah kita cover hari ni. Okay. So far ada soalan?